Welcome back. So in this video we are going to see about the restriction enzymes. These restriction enzymes, as we know that these are the enzymes that cleave the DNA into small fragments. So they cleave the DNA into small fragments. So here we get the cleaved double-stranded DNA. Now these are basically widely present in the prokaryotes. So the prokaryotes, they widely contain the restriction enzymes. Now they protect the DNA of prokaryotes against the invading bacteriophage. So when the bacteriophage invades the a prokaryote, for example a bacteria, it will try to cleave the DNA of the bacteriophage with the help of the enzymes that are known as the restriction enzymes. So here it cuts the DNA of the bacteriophage. Now the host DNA, this is methylated at specific sites, at specific bases. And now we can see that if the methylation is present in the host DNA, when the host genomic DNA, it contains the methylation at specific sites, so that the restriction enzyme, it cannot cleave that site, it cannot cleave the host genomic DNA. So that is why the host genomic DNA is not cleaved by the restriction enzyme, rather it cleaves the bacteriophage DNA. As the bacteriophage DNA is not methylated, that is why the DNA gets fragmented. So this DNA, that is the DNA of the bacteriophage, it will get fragmented, whereas the DNA of the host genome, it will not be cut by the restriction enzyme. Now we have the different types of the restriction enzymes, like the type 1, type 2, type 3. Whereas the type 2 restriction enzymes, they are most commonly used in the recombinant DNA technology, as they are very specific. For example, the EcoR1, the BAMH1, the HIND3. Now we will see the different types in exactly like the type 1, type 2 and type 3 restriction enzymes. So this type 1 restriction enzymes, they cut at 1000 base pairs away from the recognition sequence, and they require the ATP and S-adenosyl L-methionine for its functioning. Whereas the type 2, they cuts at 4 to 8 base pair palindrome sequence, and they require the magnesium 2 positive ion. So we know that this palindrome sequence, this is the sequence that reads from 5 prime to 3 prime direction. So this direction is same from 5 prime to 3 prime. The same sequence is read like GAATTC from 5 prime to 3 prime direction. And similarly from the below we will have the same sequence, the 5 prime to 3 prime, we will have the GAATTC sequence. The type 3rd restriction enzymes, they cut at 20 to 30 base pair away from the recognition sequence, that is non-palindrome sequence. So these restriction fragments, they can be blunt ended or sticky ended. So here we have the 5 prime to 3 prime and 3 prime to 5 prime direction. So this GAATTC sequence, it can be cleaved. So this is the sticky end. So here we get the G, and then we get the AATTC, and in the below we get also the G, and then we get the AATTC. So this is the sticky end. So the restriction fragments, they can be the sticky fragments, or sometimes they can be blunt ended, that is GATATC, and then we have CTATAG 5 prime to 3 prime direction. Thanks for watching.